Thank you for joining this patient-centric sampling interest group webinar. My name's Neil Spooner and I'm your host today. Today we're joined by Elham Hedayati, and Sofie Sibia of the Karolinska University Hospital in Sweden, and they're going to talk to us about opportunities and challenges in mobile phlebotomy and self-sampling for precision chemotherapy dosing in clinical trials. Before we get started, just like to take a moment to thank our sponsors, without whom we wouldn't be able to support these webinars. So thank you very much to our sponsors. And we're just opening our new sponsorship year. So if anyone's interested in sponsoring patient-centric sampling interest group, please drop me a line. So the agenda is, first of all, Elham speaking and then Sofie. Hopefully we'll have some time for questions at the end. Please could you submit your questions through the chat function and we'll do our best to answer them. With that, I'm going to go offline and hand over to Elham. Thank you.
Thank you so much, Neil. Thank you for the invite. And hello, everyone. My name is Elham. I'm a clinical oncologist here at Karolinska University Hospital. I'm also associate professor in oncology, and I'm also the co-founder of TrueDose, which is an innovative and new technology for patient-centric sampling and TDM. Today, me and my colleague Sofie will talk about the opportunities and challenges in mobile phlebotomy and self-sampling for precision chemo dosing in clinical trials. And this is the agenda for today. First of all, I will go through the breast cancer care process with you, and then I will talk about the drug development and the phase 1 designs. Shortly overview of TDM in oncology, and Sofie will take over, talk about the TrueDose trial, which is ongoing. In the end, I will discuss about the current existing microblood sampling technologies and the conclusion. Hopefully you all will be able to follow what we are saying. If not, please do chat with us and put your questions. Very shortly, breast cancer is the most common female cancer in Sweden, but also worldwide. In Sweden, we have about 9,000 new diagnosed cancer patients every year. And the care process is divided in different stages. Stage 1 is to receive the diagnosis, so the screening and the diagnosis. It takes about 4 weeks for these women, mostly women, very, very few percent of men. We have annually about 72 male patients per year. And then later on, they need to go through a treatment. The treatment usually consists of surgery and also systemic treatment. And then later on, they go into a follow-up process, which is containing mammography once a year and other checkups. In parallel with that, we are always making sure that we can include our patients in ongoing research projects and trials. And these patients, they are offered to go through a rehabilitation. When it comes to the systemic treatment, they always about 40% of the females, they receive chemotherapy, which is going about 6 months, less than 6 months, but between 5 and 6 months. And then afterwards, they continue about 80% of them with endocrine therapy that can last up to 10 years. And they also, since a few years back, it is indicated that they should also receive targeted therapy with CDK4 and 6 inhibitors. Called abemaciclib, actually. So this is how it looks like, the care process for a female breast cancer. When it comes to drug development and design of a phase 1 study, as you know, the aim is to look at the maximum tolerated dose, and it is usually based on very little number of patients, because you start always with the starting dose is given to usually 3 patients in one cohort. And if they tolerate that, then we go up to the second step and another 3 patients are included in that step, and so on, until you actually see that 1 out of these 3 develops a toxicity. And this is called DLT, dose limiting toxicity. And if they see that, they then repeat with the same dose on 3 new set of patients. And if it's observed the DLT also among this one of these 3 patients, then they go one step back, and that will be the recommended dosage. So this is how that dosage is then chosen, goes to phase 2 and a phase 3 trial, and then it's getting registered. If everything goes fine, then it is registered on that dose. So which means that this dosage that is then approved for clinical use, it is based on a very little number of patients that you see. And that's why I believe that the TDM, which is called therapeutic drug monitoring, is needed in oncology, because chemotherapy has a relative narrow therapeutic window, and if the exposure levels are not within the optimal range, then there is a risk of either lack of efficacy or severe toxicity. So you can see it's estimated that about 55% of the patients receiving chemotherapy, they are suboptimal optimal exposed to the treatment. And what does it mean? Well, it means that more than one of 3 are underdosed. And in the studies of patients with breast cancer, it's shown that lower doses are associated with decreased efficacy. And in everyday clinical practice, what we do is that we try not to dose reduce more than 30%. But it's sometimes difficult, especially nowadays when we have combination of different treatments that are given at the same time. And the group of patients that are really at risk for underdosing are those with obese patients. Unfortunately, the dose regimens are not designed for this group of patients. There is always a limit, a higher limit for how much we can give. Even though the American Society of Clinical Oncology, the ASCO guidelines, says that chemo dosing should actually be used according to actual body weight in obese patients. And my colleagues, and especially and also here from Karolinska University Hospital, we have the last 2 decades tried to see how we can improve that. So there is a way to reduce the number of underdosed patients, is to stepwise increase the dosage until there is an acceptable grade of side effect. So the trial that we had here, for example, is called Panther trial. And when it comes to toxicity, as you remember from the figure before, 1 out of 6 patients are actually in the risk of severe toxicity. And in clinical practice, we try to avoid the effect of chemo-induced tox by taking blood counts and monitor that. We make sure that if they have, for example, low white blood cells, that they get some stimulation so that they can be increased, and they are fit for fight for the next treatment. We also give anti-nausea medication, so proactively decrease that side effect, which is a very common side effect for all the chemotherapies. And another way of handling this is that we try to rearrange the dosage and the dosing schedules just to avoid severe toxicity among these patients. But this method is not so, there is not so much evidence behind that. Actually, there are no studies about that, but we have done it for many years. So there is a clinical evidence. So unnecessary toxicity has a significant negative effect on patients' quality of life. And how can we increase the efficacy of chemotherapy? Well, that has been the question, as I mentioned before. The last 2 decades, the trials and other studies suggest that increasing the dose intensity of chemo, for example, by either shortening the interval between the treatment cycles or by giving higher doses of drug individually rather than lower doses concurrently, might improve outcome. And there is a big collaboration group called the Early Breast Cancer Trial. This collaboration group that in Oxford, they usually do a big patient-level meta-analysis of the trials. In this case, 2019, they did so on more than 37,000 women with early breast cancer in 26 randomized trials. And they found that patients who are receiving a dose-intense treatment, they actually have a better outcome in terms of recurrence-free survival and death mortality in breast cancer compared to conventional non-dose intense treatment. The same group in 2012 did an analysis and saw that the chemo is better than non-chemo. Together with this meta-analysis in 2019, we can conclude that the combination of anthracycline treatment and taxane-based treatment reduces the risk of breast cancer mortality by 40% compared with no chemotherapy during the first decade for patients with early breast cancer. The same group did exactly the same, but for endocrine treatment, in this sense, tamoxifen, and they've shown that if you receive tamoxifen 5 years compared to no tamoxifen in breast cancer patients with endocrine sensitive tumors, the 5-year, 10-year, and 15-year survival, you see that the recurrence-free and the survival is much better for those who receive 5 years tamoxifen compared to no tamoxifen. And another study, the Atlas trial a few years ago, showed that not only tamoxifen is better than nothing, but if you give it 10 years compared to 5 years, it is better. So the tamoxifen matters, and the 10 years tamoxifen is better than 5 years tamoxifen. So this is what we tried. There was a change in guidelines. So since then, we are trying to make sure that these female patients try to have them on the treatment as long as possible. However, with these long-term treatments, it comes also some challenges. So in these 10,000 women who were in these tamoxifen trials, in these trials, we saw that 20% of them didn't complete the treatment because of side effects usually. And in real life patients, we know that, and especially in Sweden, because we have registry-based studies, we know that 50% of the patients, they don't complete the 5-year course of treatment. And that is usually due to side effects. And it can also be due to the lack of shared decision-making between the oncologist and the patient. And there are not so many studies, any trials anymore for these patients to be enrolled in. So currently, there is no gold standard available to assess adherence for this very important treatment. And the indirect method is more widely used and based on pharmacy prescription refills and patient administration questionnaires, or for example, for us here in Sweden, we have a very good renal quality registry. We can look at that. But of course, direct methods such as measuring the level of the drug and the exposure in the blood is much more precise, but more expensive, and it needs to be routinely implemented, and we have not done that yet. When it comes to targeted therapies, it is also the new era for precision medicine, and within breast cancer, we have lots of new treatments coming in, and this is the data from the PALAS trial from a few years back. It's a CDK inhibitor for adjuvant setting, and here you can see that about 42% of the patients, they discontinued the treatment during the trial mainly because of the side effects. And they themselves concluded that there is a need of new methods to be implemented so that we can make sure that these patients are adhered to the therapy that we recommend. And then you can question, why is TDM not routinely used within oncology? Well, most cancer-specific therapeutics are still used as a one-dose-fits-all approach, and dose optimization often is overlooked and remains a crucial and significant part of precision medicine in cancer treatment. And in the current era of the precision medicine in oncology, treatment is mainly tailored to the individual tumor characteristics to optimize the therapy outcome. However, the dosing of commonly used cancer-specific drugs has yet to enter this era. And this many of you know, but there are different key factors explaining the variability of exposure. Within oncology, we believe that only body surface area is the main important, and we choose the dosing either by weight or body surface area or flat dosing. But as you can see in this figure, there can be other factors that can explain. It can be, for example, co-medication, it can be organ function, the lifestyle with the smoking and the alcohol intake, or the genetic factors, of course, and the sex, of course, and the age, but also the illness itself. It can be that throughout this time, when if it's a metastatic setting patient, the illness can progress and cause that the exposure levels vary, but also other factors that we don't know today. So it is impossible to estimate the exposure only with the BSA, since other factors also contribute to exposure level. And how to overcome this challenge? Well, it is to stop guessing and start to measure the exposure. And one can question, okay, why don't you do that exposure measurement? Because for every drug, after each intake, we would like to know the absorption, the distribution, and the elimination. And to do so, we need to measure it in several times. And to do so, then we need to have the patient in-house and hospitalized, and it's not cost effective at all. That's why my colleagues and me, we are still very confident with the BSA concept when it comes to chemotherapy, because it is more convenient. A colleague of ours in the Netherlands for 20 years ago did a summary of the most common chemotherapy agents used in beginning of 2000. And as you can see here, the variability between individuals is high. It is everything from 25% up to 70%, even, and the variation in the body surface area was 15% only. So this figure shows how important it is to actually measure the exposure, so that we have the individual difference in the absorption and distribution and metabolism and excretion. So some of you know that we have pharmacogenomics as a tool today, and that's very good. However, pharmacogenomics doesn't still give us the end outcome result. It doesn't give us how much active metabolite it is in the bloodstream. So pharmacogenomic is good, is very good to have, and it's a complement to TDM, according to me. So in summary, chemotherapy in oncology will always be the backbone treatment, regardless with surgery, radiation, or other targeted therapies. And it can be given before surgery, after surgery, or as different lines in a metastatic setting. And the challenge with chemo is that they have a narrow therapeutic window, and dosing must be precise to avoid underdosing or overdosing. High inter-individual variability in patient response due to differences in absorption and distribution and excretion. And TDM involves measuring drug level at specific time points to tailor dosing to each patient need. And this is especially critical for drugs like chemo, where standard dosing is usually based in for body surface area, and it doesn't suit all patients due to varying metabolism and drug exposure. So the goals of TDM is to ensure optimal drug exposure for maximum efficacy, minimize toxicity by adjusting doses based on real-time measurement, and improve patient outcome through personalized data-driven treatment. So, and with that, I give the word to Sofie.
Hear me? Yes. Thank you, Elham. And I will present to you the TrueDose performance trial from a mobile phlebotomy perspective. And the aim of the trial in this study is to develop a better tool for medical dosage administration to reduce the side effects while still maintaining a high medical effect. And the trial setup that we've been performing, it includes that the patient gets their drug intake at site as part of their standard of care procedure. And the patient will then be followed up at 2 sampling occasions after 3 hours and after 24 or 48 hours, depending on the type of medical administration they've been given. To evaluate the new patient-centric self-sampling tool that is done by a capillary sampling method, both a capillary blood collection and an intravenous blood collection is performed. So the drug administration and the first follow-up collection is done at the clinic, whereas the 24 or 48-hour collection is offered to the patient as a home visit. And a challenge that we identified for this study design is that the follow-up sample collections are in very close time to the drug administration that the patient's been given very recently at site. And from previous experience from other studies, it's expected that patients drop out, leading to high causes of missing data and protocol deviations due to adherence issues. So to overcome this identified challenge, we introduced at-home visit solutions to patients to provide them with greater flexibility. Both the standard intravenous sample and the capillary microsampling is then performed at the patient's home by a healthcare professional, ensuring compliance with the protocol and to the timelines. And the outcome of this approach is the reduced patient burden and for the patients to have a choice, allowing them to choose their preferred follow-up sampling location and method. And as the home visit can be scheduled throughout the week, including weekends, this increases the inclusion potential as drug administration visits can be scheduled at the site Monday to Friday, also reducing the site burden to allow them for a full week patient visit scheduling. So when performing home visit solutions, it's important to consider all aspects needed to be in place for a successful data collection. The at-home visit is the central part, and all the other building blocks must interact with each other, all equally important for the home visit to be performed as needed by the protocol. Project management before the project start to understand what is to be performed, what type of collection are to be done, and how will the patient receive the material. For this performance study, the patient received the capillary kit after the drug intake at site to take with them home. In other scenarios and for future work, the patients may need to have a logistic solution for them to get the self-sampling kit directly shipped to their home. And how do the logistic solution look after the samples are taken? And for the mobile phlebotomy service part, a major part is the scheduling of the healthcare professional. That is really important, that the right qualified person is at the right address, at the right time, at the right day. And when all of these building blocks are working together, there is a successful visit procedure performed, allowing for home visits and increased patient flexibility and reduced patient burden. Let's pass on back to Elham.
Thank you, Sofie. Yes, so the next slide is very shortly an overview of current existing microblood sampling technologies. This is a figure that I have taken from this review article, and you can see here are different techniques. This is the dry blood sampling, and there are the volumetric absorptive microsampling and the automated microsampling devices. And what they have in common, both of them, that there is this hematocrit issue variability, and it can affect the accuracy, especially in patients with low levels of the analyte that we are going to measure. And when it comes to DBS, there is also the sample preparation time that requires longer sample preparation, impacting the result turnaround. When it comes to VAMS and the other techniques, limited validation still out there, but I know that there are trials going on, and it needs more. There is a need of more studies with these techniques, but hematocrit also here has an impact on the results. Let's go see. So the application of the microsampling technique can be on volume limited application, such as in the animal models or the neonatal and pediatric studies, where less invasive sampling is needed, and or omics-based biomarker research, where high throughput analysis of biomarkers can be done. It can also be used in forensic toxicology to enhance the ability of detection and quantification of the drugs or toxins. In sport anti-doping, it can be used, and it can also be used in precise monitoring of drug levels in TDM. But there are limitations with today's microsampling techniques. One is the sample stability. The transport and storage are likely to affect the results reliability. Then later on, the analytical sensitivity due to low collecting volumes. And the third is this hematocrit effect, where the levels can significantly impact the accuracy of sample volume measurement and leading to inaccuracies in quantitative measurement of analytes. And if you look at the standard IV patient flow compared to the dried blood spot patient flow, the standard care phlebotomy, the patient, if I wanted today do the TDM in oncology in my clinic here, then I need to make sure that that method is up and running at our lab. So I need to make a call to see when they are available to collect, for the patients to be coming to the lab for collecting blood. Then that blood needs to be transported to the lab, and then it is prepared and analyzed, and I get the full results maybe within about 2 weeks or a month or 6 weeks. So it's a long time process. During this time, the patient will start taking the treatment again. And if you look at the dried blood spot, it's the same process. I need to make sure that they have the method up and running, and then the patient can take the blood sample at home and post it. And then when it comes to lab, the extraction process will start, and then it will get analyzed. So what we have done with the TrueDose technologies, and the technology is a liquid-based, and it actually makes it removes the process of the lab work. So you can see here, compared to the other technologies, when the patient can take the sample at home, it is transported by post to the lab, and when it comes to the lab, it can be analyzed directly, and the answer is back to the physician quite fast. So the difference is liquid-based, and if you are interested to know more about the technology, please visit our homepage. Since I'm an oncologist and not an organic chemist, so I will not go into that discussion with you. So the products that we have in pipeline is the chemotherapy, the endocrine therapy agents, and the targeted therapy agents that we are working on, and we are hoping that we will have the first CE mark product by beginning of next year. With that, I would like to conclude the talk that our case study with the TrueDose trial demonstrates how measuring chemotherapy exposure through patient-centric sampling can drive a more effective, safer, and personalized cancer treatment. And there are many different microsampling techniques available nowadays, and they should be evaluated and used in different patient cohorts. So, thank you. I think I'm in time, 29 minutes.
You're beautifully on time. You're very early. We've got lots of time left. Thank you so much.
Thank you.
Sofie and Elham. So if people don't mind submitting their questions through the chat function. I see there's some starting to come in, so I'll just read these out. The first one's from Jeff. I think it's more of a comment than a question. He says, hematocrit-induced sampling volume bias is eliminated in Tasso, Mitra, and Capitana B devices. Further, with proper sample preparation workflow, the impact of hematocrit on analyte recovery can also be eliminated. So do you have any comment on that at all?
No, I say thank you for that information. Very good.
Anthony is saying, got to be a place for population PK into future study design. So is that something that you're looking at, you know, sparse sampling and population PK models?
Yes, we are going to run that in our TrueDose trial by the end of the study. We need to have, we are still missing about, I think, correct me if I'm right, Sofie, I think we are like 60 patients left to include in that trial.
Correct.
So you still don't have outcome data from your trial yet?
No, it's still ongoing. So hopefully next time you invite us, then we will present the data from the TrueDose trial.
Yeah, that would be really good to see. Are you also doing patient questionnaires to find out what they think?
Yeah, of course. We do patient questionnaires to look at the symptom measurements, but we also look at the health economic part of that. So yes.
Question from Karen. Is this a finger prick or an upper arm type device?
A finger prick.
Okay. But so you've not included some upper arm collection devices for comparison. It's just the finger prick.
It's a finger prick with the TrueDose technology compared with IV sampling.
What volume do they need to collect with the TrueDose?
50 microliters.
Okay, so it's not too much.
No.
Because some tests need quite large volumes from a finger prick, which is not reasonable, whereas 50 microliters is a reasonable volume. And is that then shipped as whole blood?
Yeah, the whole tube is, I can show you the, if we go back, I don't know if we can go back. You see the tubes?
Yes.
Yeah, so you activate the kit by pressing this cap on it, and then you take the blood, you press the blood down 50 microliters, you close it, you shake it, and you send it by post back in the package is the one behind. So it's liquid based, and everything is stabilized directly so the patient is both a sampler but also a BMA in one.
So what's your mechanism for capturing both the time of the sample and the identity of the person that the sample was collected from, just to make sure, because they're obviously two very important components.
Yeah, they will be labeled and there will be a QR code like the ones that we had in COVID, during the COVID. So it will be a technology behind, of course. So the patient needs to use an application.
Okay.
Yeah.
And that helps capture the time as well?
Yes.
Okay, cool. There's a question here from Shelby. Are you measuring the cyclophosphamide active metabolites? If so, how are you stabilizing the sample to measure the 4-hydroxy cyclophosphamide?
Yes, we do. And I would say that for that, we need another lecture because my colleague and co-founder, Per Rydberg, he has actually invented a technology how to measure that. So, but I think we need to have another lecture for that, and so that he can talk about how that technology is working. But yeah, so he has a technology, patented technology that he's looking at.
Okay. Another question from Karen. Would love to know more about how the lab processes the sample. Is this a specialized lab or a routine clinical lab?
It's the LC-MS apparat we need. So
okay.
Yeah.
And it's an assay that you were already using, or did they have to develop it specifically for this sample type?
It can be any assay that they have, but usually since these are not tested today, TDM in oncology is not up and running, so we need to set up all the method for each sample, for each drug.
Okay.
Yeah.
It's LCMS. That's good to know. So,
yes, we do.
okay. Do you include? I'll just read it out so everyone can see it. I don't know whether everyone can see all the questions. Do you include any liver function tests, full blood counts, renal function on these patients using this method?
Not right now. We are a startup, so we don't have. So currently we are just focusing on TDM, but of course that is in our pipeline to do so, because it would be making the life for these patients much more easier. Yes, so in future. But in the IV sampling, of course, we are doing that. So in our IV samples, we look at all those.
Okay. And Karen says, sorry, very interesting and a great system with loads of potential. So thank you, Karen. She's been very complimentary there.
Thank you.
Well, Karen's our next speaker in December. So I think it's her. So this is currently just a kind of prototype system. You know, how easy is it going to be to scale this up?
Well, it is not a prototype because we are running the trials now. It is manufactured in Sweden. Is it called? So there is a scale up. We have already scale up in our mind. So we are collaborating with many partners here in Sweden.
So is the intention to spread it to other centres in Sweden, and what about elsewhere in the world?
Yes, and elsewhere in the world. Yeah. So we know that the interest of our technology is quite high within oncology. So from the beginning, we were thinking, we have been planning for scale up.
So Jeff has a question here, and he's obviously spotted something. He's been zooming in on some photos or something. I know Jeff has got a real good eye for detail. He says, what is the purpose of the stainless steel beads?
Once again, I'm only a simple oncologist, but I will try to say what my colleague Per, who is actually the analyst here, it is for the protein precipitation.
Okay.
But please email us. Very good question. I cannot answer all your questions. Email us, and we will get back to you so you can get detailed response.
Are you able to put your email address in the chat function?
Of course. Yes.
Okay. Yeah, that would help so people can connect with you. Yeah, well spotted, Jeff. I didn't even spot that. So you've not got outcomes data. How long are the samples actually stable for? Do you know?
In the TrueDose technology, they are stable up to 14 days and hopefully more. We are running tests. The IV samples, we freeze them directly. We prepare them and we freeze them in the freezer until we will analyze them.
Whereas the TrueDose are at ambient room temperature, yeah?
Yeah.
For 14 days?
Yeah.
That's pretty cool. Anthony has a question. Interested to learn more about those tubes and availability of the collection device. He's wanting to know how generally are they commercially available, I guess. And Karen wants to know too.
Yeah, you know what, guys? Email me and I will get back to your questions.
It looks like a lot of people want to go shopping with you.
Yeah. Let me just send you my email address here so you have it.
So Azita's just said they're not yet commercially available, but that's Elham's email address. Is everyone able to see that? And Azita said we're in the middle of CE marking process.
Yeah, she's the CEO of the company.
And thumbs up from Jeff. So I assume that means he can see the email address. And Azita says we will launch during 2025. So Shelby, a lot of information out there for these drugs will be in the plasma rather than whole blood. Do you convert these values so that you can compare with the literature?
Yeah, of course. Absolutely. it is a part of our CE marking process for each product.
Okay, cool. Hi everyone from Azita, a thumbs up from Anthony. I've run out of my questions. Oh, here we go. Karen's going, do you have any qualitative data to share from your patients?
Not right now, but we will have a publication soon.
Okay. And Anthony's saying good timing is potentially have a study that we need to consider patient-centric sampling. So you're getting people excited here. It's
yes,
good.
I see that. Yeah, but I think that, I mean, really, I feel like the whole idea, because maybe I didn't mention it very clearly, when we started our TrueDose study in 2017, after 1 year, and I'm a quite good PI, so I usually try to include my patients in all the trials that we have up and running. But this study was impossible. It was impossible to include patients in this trial. And this was because the time of sampling is so important. So even if the patients wanted to be included, I couldn't include them because the labs are not open for the IV sampling. The labs are open between 8 to 3 o'clock, and they close. And most of the labs that time, if they were not university hospital labs, they were not taking research samples anymore at all. So it started there that I forced Stockholm to implement that they could collect research samples in any laboratory, even close home to the patient, during their opening hours. Then we saw that the inclusion got up a little bit, but not so much. So 2018, me and my colleague Per, we were in the same research group, we came up with the idea to do a capillary. We were thinking, how can we increase? That was really our need. We needed to have a tool to increase the inclusion in our study. And that is how the idea of TrueDose actually came.
Yes.
And we got innovative, we got money from the Swedish organizations, and now 2024 we are sitting here and we have a technology almost CE marked.
So with that CE marking and everything, you've done a bridging study not only between blood and plasma, but between venous blood and capillary blood?
Yes, so we will measure. We always compare plasma with whole blood IV and plasma whole blood capillary, and then whole blood IV versus whole blood capillary. Yeah.
So what are your thoughts on the number of samples that are needed in order to do that bridging?
I think 40 samples is enough, because 40 samples will generate different comparisons. So 40 samples are enough.
And that's, so there was a PBSS event last week that I was co-hosting out of California. And Brian Booth from the FDA for pharmaceuticals was saying that seems to be the kind of number that they're receiving for pharmaceutical registration for bridging between blood and plasma and capillary and venous. So I assume you want to get them at different concentrations as well.
Yeah, of course, because it's always a higher concentration in the plasma compared to whole blood, but you need to look at the ratio. So yeah, I don't know if I answered your question, Neil, but
no, you did. Aziza's got a question. Does the absence of anticoagulant have no effect on the quantification of the test compound when the sample is taken from a finger prick? So obviously your control blood for your calibrants and your quality control samples has an anticoagulant, whereas your collected blood doesn't, or is there anticoagulant in that tube?
There is, I think. I let it be, Aziz, to answer your question, but I will take it home. And if you're interested, email me and I will get back to you. I should know that question, actually.
He's answered. Azita said they're coated, so I assume that means they're coated with an anticoagulant.
It's good that I have her behind the scenes.
And Anthony suggested this is a good discussion topic for PCSIG. Yes, absolutely. We love these kind of discussion topics, and that, that's what our community is for. So
Thank you.
please launch these discussions within the LinkedIn group, or if there's enough interest in a particular topic, we can launch a new work stream to get stuff done. So
excellent. And please follow us on LinkedIn and our homepage. I'm not so operative myself, but I know the team behind are working very hard every day. So contact with Azita and the company for more detailed information.
No, you've created a lot of excitement today. Karen says, have you looked at any tumor markers using these devices?
No, but we are planning to do so. It's actually very high interest from my fellow researchers here at KI. It's in pipeline, but we have to focus on one thing right now, and that's TDM. And next will be other markers and blood counts and liver function, etc.
Karen says, thank you, excellent presentation. But it sounds like there might be a few people out there willing to try and help you.
Yes, we are very happy to collaborate with anybody who is interested to develop this technique further.
Cool. So, any more questions from out there? Okay, well, I'm just going to advance the slides to my slides at the end, if that's okay. So this webinar is being recorded, and in a few days' time, it will be available through both the evidence base, through the Patient-Centric Sampling Interest Group website, and through the Patient-Centric Sampling Interest Group YouTube channel, which if you've not been there and had a look, please go and have a look and follow that. So that should be uploaded in the next few days. We've got some more webinars coming up. So November the 12th, so in just a few weeks' time, we have a presentation from the PCSIG board, just telling you a little bit, giving you a bit of a chance to meet the board and telling you a little bit about what we've been doing this year and a brief outline of what we're looking to do next year. And then in December, in her Santa Claus outfit, we'll have Karen, who's been asking a lot of questions here. She says, yikes. Paradigm shifts in phlebotomy, providing reasonable adjustment for underserved patient populations. So I'm very much looking forward to that presentation in December to bring us to the end of the year. So with that, just like to remind you a little bit about the Patient-Centric Sampling Interest Group. We are a free-to-join group. We have a lot of content now on our website, which you can get with that QR code on the left. We have a lot of content and a lot of discussions and announcements through our LinkedIn group. Please feel free to join. It's a closed group. We just do that to stop recruiters wandering in and people that might bother people, but use the QR code on the right-hand side, or contact us using that email address, contact@pcsig.org, or email me, neil@pcsig.org. And with that, I'd just like to, again, thank our presenters today, Elham and Sofie. This was absolutely brilliant. What a great discussion we've had, great presentation. And obviously we need to thank our partners and our sponsors, without whom we just couldn't afford to do what we're doing today. So, and thank you, the audience. Thank you for providing such great discussion and such great questions. It really has been a lot of fun today. So thank you, everyone. And see you soon. See you in a few weeks' time for the next webinar.
Thank you so much for the invitation and for listening.